There is moss within walking distance of where you are sitting, probably on a stone wall down the road, or in the cracks of an old set of paving slabs, or on the shaded side of a tree, or at the edge of a roof tile that has been there long enough to forget it was ever clean. Moss is one of the most common things in the world, and most people, if they think about it at all, think of it as a surface, a kind of slow green decoration that grows where nothing else can be bothered to grow. In every square metre of healthy moss, there are roughly 2 million animals. You almost certainly have not seen one. They are about 0.5 millimetre long, 8-legged, with a body that looks, under a microscope, like a small barrel that has decided to grow shoulders. They walk slowly. They eat the inside of plant cells through a pair of tiny mineral spears in their mouths. They are not insects, not arthropods, not microbes. They are their own thing, their own phylum, biologically speaking. The formal name is tardigrade. Most people who know them at all know them by the affectionate name water bear. The water bear has been on Earth for more than 500 million years. It has lived through every mass extinction the planet has staged, 5 of them, including the one that ended the dinosaurs. It will probably live through the next one too. And in the last 18 months, 3 separate research groups on 3 separate continents have published findings about it that have, quietly, unsettled some basic assumptions in biology. Most of what people say about this animal turns out to be wrong, or at least misleading. It is not indestructible. An active water bear is, in fact, almost embarrassingly fragile, and you can kill one with the edge of your fingernail. It does not live in space. In 2007, it became the first animal in history to survive open space for several days, but only while dehydrated and effectively unconscious, which is not the same thing as living there. The water bear is famous for the wrong reasons. The right reasons are stranger. In a moss sample collected on a mountain in central China, researchers recently found a gene that this animal appears to have taken from a bacterium, possibly 500 million years ago. The gene makes a kind of pigment normally found in plants and fungi, the deep red of beetroot, the purple of dragon fruit. The water bear uses it to clean up the chemical damage caused by radiation. A small animal defending itself against extreme radiation with a piece of plant chemistry encoded by a gene borrowed from a bacterium is not how biology textbooks usually describe how an organism solves a problem. It is, however, accurate. In a laboratory in Iowa, a different protein from the same animal is currently being tested as a way to protect healthy tissue in cancer patients during radiation therapy. The protein is being delivered into mice and into human cell cultures inside tiny lipid bubbles, and the early results suggest it works. The next phase is people. And in a crater on the Moon in the region called Mare Serenitatis, several thousand water bears have been sitting in dehydrated silence since April 2019, when an Israeli spacecraft called Bereshit crashed during landing. They were carried up there as part of a long-term archive of life on Earth. Whether any of them survived the impact of the crash is unknown. They cannot revive themselves. The Moon has no water. You can see the Moon most nights without effort. You probably saw it yesterday. The water bears in that crater have been there for nearly 7 years. If what you're listening to feels worth being made, the subscribe button does more for a channel like this than almost anything else. And in the comments, here is something genuinely interesting to know. Is there a patch of moss somewhere near where you live? On a wall, a stone, a tree, the edge of a path? The answers people give to that question are surprisingly varied, and they tell me a great deal about who is listening. I read them. Tonight, somewhere on this side of the planet, the moss is going to get wet. Out of the millions of small animals in it, a few will start to walk. They will not know about the laboratories. They will not know about the moon. They will not know about you. They have not, generally speaking, ever needed to. The moss has been wet for about an hour. Somewhere in it, perhaps a few centimeters in from the edge of a paving stone in a garden, perhaps on the north side of a stone wall outside a village in the Cotswolds, or in upstate New York, or in a damp courtyard in Kyoto, an animal that has been folded into itself for several weeks is beginning to unfold. The water has reached its body. It has crept in through the cuticle the way water creeps into a dried sponge, slowly at first, then everywhere at once. Cellular structures that had been compacted into something glass-like are loosening. Proteins that had locked into protective gels are dissolving back into the cytoplasm they came from. The metabolism, which during the dry weeks had been running at something close to 1/10,000th of its normal speed, is climbing again. First into the realm of the measurable, then into the realm of the active. Around the 20th minute after the first drop of water arrives, a leg moves. Around the 30th, another. By the time 40 minutes have passed, the animal is walking. Walking, for an animal of this size and this design, is a specific thing. It has 8 legs, arranged in 4 pairs along its underside. Each leg ends in a small cluster of claws, 4, 6, sometimes 8, depending on the species, and the claws hook into the texture of the moss the way a climber's crampons hook into ice. The animal moves with a slow, deliberate, slightly bumbling gait that has earned it, in several languages, the same affectionate comparison. A bear. A small bear on a long, slow, methodical walk through a forest of green threads. The species we are watching, if we want to be specific, is most likely Hypsibius exemplaris. It is one of the most common terrestrial tardigrades in the temperate world, and it is the species most often used in laboratories, which means we know more about its body, its genome and its habits than we know about almost any other water bear on Earth. The animal is about 0.5 millimetre long. Its body is mostly transparent, with a faint amber tint concentrated in the gut, where the algae it ate yesterday are still being digested. The most interesting part of the animal is hidden in its mouth. Retracted into a sheath of muscle, 2 thin spears made of aragonite, the same crystalline form of calcium carbonate that gives mother of pearl and certain seashells their hardness, wait there fresh and sharp. They are called stylets, and they are, for an animal this small, terrifying. When the tardigrade encounters something it wants to eat, a soft moss cell, a strand of algae, in some species the body of another small invertebrate, the stylets shoot forward, puncture the cell wall, and retract. The pharynx then pumps the contents of the cell into the gut. Every time the animal molts, which it does several times in its short active life, the old stylets are shed and a fresh pair is grown to replace them. The animal carries in its mouth a constantly renewed pair of mineral weapons. It has been doing this for over 500 million years. This morning, the cell wall the animal is going to puncture belongs to a moss leaflet barely thicker than a soap bubble. The puncturing happens, the contents are drawn in, and the animal moves on slowly towards the next leaflet. Somewhere in the same patch of moss, possibly within a few millimeters, there is a nematode, a tiny eel-like worm that also lives here. Some nematodes are harmless to a tardigrade, some are not. Certain species prey on water bears, piercing them, draining them, leaving the empty cuticle behind. Fungi can infect them, slowly hollowing them out from the inside. Protozoans latch on. Other tardigrades, in fact, hunt smaller ones. The tardigrade in this moss patch is, in any reasonable sense, prey. It will, on average, live for a few weeks. Most members of its species do not die from anything dramatic. They die from being eaten, from being parasitized, from a fungal infection, or from a drought that arrived too suddenly to allow the body time to prepare for it. It is at this point that something needs to be said, because the popular story has gone, for a long time now, in a different direction. The popular story is that this is the toughest animal on Earth. The popular story is that it can survive being boiled, frozen to near absolute zero, crushed at 6 times the pressure of the deepest ocean trench, blasted with 1,000 times the radiation that kills a human, and shot into the vacuum of space and brought back alive. All of these claims are, in the right context, true. But the right context is doing a great deal of quiet work in that sentence, and the quiet work is the actually interesting part. A water bear in its ordinary walking, eating, drinking state, what biologists call its active state, is not tough at all. You can kill one with a fingernail. A drop of soap can kill it. A few minutes in the wrong kind of water, too salty, too acidic, too hot, can kill it. The animal we are watching this morning in this patch of damp moss is, by any reasonable standard, fragile. The legendary survival, the freezing and the boiling and the radiation and the vacuum, all happens in a different mode. It is not the animal's default mode. It is, more accurately, the mode the animal switches into when the ordinary world has stopped being survivable, when the moss begins to dry, when the temperature climbs past what the body can manage, when the oxygen runs out or the water turns to ice. The tardigrade does not endure. It withdraws. The legs come in. The body folds. Water is expelled, almost all of it, sometimes more than 99%. What is left is a small, dry, barrel-shaped object that bears the same relationship to the original animal that a folded glider bears to a flying one. It is called a ton. The ton is what does the surviving. The ton is what the famous experiments are done on. The ton is what was sent into space in 2007, and what was packed into the Lunar Archive in 2019, and what was placed in a refrigerator at fractions of a degree above absolute zero in 2021. Inside the ton, the animal is no longer, in any conventional sense, doing anything. Its metabolism has dropped so far that for many years, biologists could not detect it at all. The cells have entered a state of suspended chemistry. The DNA has been packed and protected. The proteins have folded into shapes that hold the body's structure together in the absence of the water that would normally do that work. And then, when the conditions improve, when the moss is wet again, when the temperature falls back into the survivable range, when an ice age ends or a drought breaks, the ton is rehydrated and the animal walks out of it. The same animal, the same individual, picking up more or less where it left off. The trick, when the popular story is examined carefully, turns out not to be that the water bear is unusually hard to kill. The trick is that it can, when it needs to, leave its own life and come back later. The fragility and the immortality belong to the same animal. The animal has, in effect, a switch. The switch has been there since at least the Cambrian period, which is to say since long before there were trees, or insects, or anything resembling the world the animal now lives in. The switch is older than almost everything in the room you are sitting in, including the room itself. The animal in the moss this morning is using equipment that has been refined over a span of time so vast that there are very few honest ways to describe it. There are, in fact, ways to describe it. The most useful ones are dates. In the late autumn of 1773, in a parsonage in the small Pomeranian town of Quedlinburg, a Lutheran pastor named Johann August Ephraim Goethe was looking through a microscope. Goethe was a man of his time, devout, methodical, fascinated by the minute details of God's creation, and possessed of the patience that examining pond water with 18th century optics required. The microscope itself would have been a brass and wood affair, lit by a candle reflected through a small mirror with a single ground glass lens that distorted everything at its edges. Goes had been examining samples from the local moss and damp soil for some time. On this particular evening, he saw something he had not seen before. A small creature, 8-legged, slow, with a strange ambling walk that reminded him of a very small bear. He wrote it down in German, in a private register that would later be published as part of a longer work on freshwater microorganisms. He called it the Kleiner Wasserbär, the little water bear. This was the first time the animal had a name in a language anyone living today still speaks. It was, in the official record, the moment the species entered human knowledge. The animal had been on Earth, by that point, for somewhere between 500 and 600 million years. Goes was, in evolutionary terms, the latest of late arrivals to the room. 3 years later, a Catholic priest and naturalist in northern Italy named Lazzaro Spallanzani, already famous for the experiments that would become the foundation of modern biology, picked up the same animal under his own microscope. Spallanzani gave it a Latin name. He called it il tardigrado, from the Latin words for slow and step, on account of the unhurried way it moved. That name, slightly modified, is the one the animal still carries. But Spallanzani did something more important than naming it. He noticed that when the water on his slide dried up, the tardigrade did not die. It folded into itself, shriveled, and stopped moving. He waited. He added water again. The animal unfolded, rehydrated, and walked off across the slide as though nothing had happened. Spallanzani wrote down what he had seen and, with the careful caution of an 18th century scientist, did not quite believe it. He repeated the experiment. It happened again. What Spallanzani had stumbled into, without any framework to understand it, was the first documented observation of cryptobiosis, the suspension of life processes that the water bear performs when its world dries up. It would take another 2 centuries before anyone could begin to explain what was actually happening inside the animal during those dry intervals. Spallanzani did not need the explanation. He only needed to record what he had seen, which was that a small animal had, in front of him, twice now left its own life and come back. Both of these men, Goese and Spallanzani, are people we know about. They had names, addresses, professions, surviving correspondence. They worked in a Europe of postal services and printing presses and academies, and the records of what they did and saw have been kept and studied. The tardigrade they were looking at had no such records, but it had a history, and the history is much, much longer than theirs. Most of what we know about the tardigrade's deep past comes from 2 sources working together. The first is fossils, of which there are very few. Tardigrades are soft-bodied, microscopic, and live in habitats, moss, leaf litter, freshwater sediment, that almost never preserve a body well enough for it to survive into the geological record. The fossil tardigrades that do exist are nearly all preserved in amber, the fossilized resin of ancient trees, which sometimes traps small organisms in transparent gold and holds them for tens of millions of years. The earliest unambiguous tardigrade fossils we have come from Cretaceous amber, found in New Jersey, about 90 million years old. There is one from Dominican amber in the Miocene, somewhere around 20 million years ago. In 2024, 2 more fossil species from Canadian amber were redescribed using modern microscopy, pushing the resolved fossil record a little further. None of this comes anywhere close to the actual age of the lineage. For the actual age, we have the second source, the molecular clock. By measuring the rates at which certain genes accumulate small mutations, and by comparing the genomes of living tardigrades with those of their nearest relatives, the velvet worms, and through them the arthropods, biologists can estimate when the tardigrade lineage split off from the rest of the animal kingdom. The estimate sits somewhere in the late Precambrian, somewhere around 540 million years ago, possibly earlier. This places the origin of the tardigrade body plan in or just before what is sometimes called the Cambrian explosion, when most of the major animal groups alive today first appear in the fossil record. The water bear was already among them. It was already small, already eight-legged, already crawling slowly through whatever shallow water or damp surface it had found. 540 million years is not a number the human brain processes well. It helps to put a few markers along it. When the tardigrade lineage was already established, there were no land plants. The continents were bare rock. The first true forests would not appear for another 200 million years. The first dinosaurs would not appear for another 300 million. The first mammals, the first primates, the first creature anyone might recognize as a distant ancestor of a human being. These were all, at the moment of the tardigrade's origin, somewhere between 300 and 500 million years away in the future. 5 mass extinctions have happened since the water bear first appeared. The end Ordovician, around 445 million years ago, the late Devonian, around 370, the end Permian, around 252, which killed roughly 9 in 10 species on Earth, the end Triassic, and the end Cretaceous 66 million years ago, the one that ended the dinosaurs. Each of these would have taken out in some places most of the local tardigrade populations. Each of them the lineage as a whole walked through. Whatever it is the tardigrade does, it has been doing it long enough to have outlasted 9 in 10 species on Earth 5 separate times. The lineage's nearest known ancestors are a group of extinct soft-bodied animals called lobopodians, which lived in the Cambrian seas and are known mostly from a few extraordinary fossil sites: the Burgess Shale in British Columbia, the Chengjiang in China, a handful of others. Lobopodians look, in the fossil record, like worms with stubby legs. They were not common animals. The seas of the Cambrian were dominated by stranger and larger creatures, the trilobites and the early arthropods and the great soft predators with names like Anomalocaris. The lobopodians were a side branch, small and slow and easy to miss. From them somehow came the velvet worms, the arthropods and the tardigrades. A 2023 phylogenetic study at the University of Bristol reconstructed the most likely picture: an ancestral tardigrade with a vermiform body, lobopod legs ending in claws, and the cuticular structures around the mouth that would, in time, become the stylets the animal still uses. The ancestor was already, in essence, the same animal. Smaller and stranger details would change. The basic design would not. Something else worth noting about the design: the tardigrade body, in modern morphological terms, corresponds to only a single segment of the body of an arthropod, specifically to the head segment. The water bear is, in a structural sense, an animal that consists entirely of what other animals carry as their head. The legs and the trunk and the gut all sit, biologically, on a body plan that an insect or a crab uses for the head alone. Whether this is the result of extreme evolutionary miniaturization, or whether the tardigrade lineage diverged so early that it never developed the rest of the body that other panarthropods went on to build, is still a question biologists argue about. Both possibilities are strange. What is not in doubt is the persistence. The tardigrade has been on Earth for longer than every other animal lineage anyone is going to mention in a normal conversation. It has been on Earth longer than the ancestors of insects. It has been on Earth longer than the first vertebrates with proper jaws. It has been on Earth longer than there have been ferns. The bird outside, the dog asleep on the floor, the human listening to this. All of them are recent. The water bear is the older sibling who has watched all of them arrive. Goes, in 1773, sat in front of his microscope and, with the polite German formality of the era, gave the animal its first name in any modern language. He had no idea he was naming something that had been waiting to be noticed for over 500 million years. The animal, on the other side of the lens, had no idea it was being noticed. It was doing what it has always done. It was looking for a piece of damp moss and a wall it could climb up and a small soft cell it could open with its mineral spears. The world above the slide was not its concern. And then one afternoon, in some patch of moss like this one, the water begins to leave, not all at once. Mosses dry slowly. The damp shaded patch on the north side of a stone wall might lose its moisture over the course of a long warm afternoon, the way a wet shirt dries on a line, first the surface, then the deeper threads, then the tiny pockets between them where, until very recently, an entire small ecosystem was going about its business. The tardigrade in this particular pocket has perhaps 15 minutes. It might have an hour. It might, if conditions are unusually favorable, have most of an evening. What it does not have is a choice about what comes next. What it does have is a sequence. The sequence has been refined over 500 million years, and the sequence is what allows the animal to step out of its own life for as long as the world remains unsurvivable, and to step back in when, eventually, it is survivable again. For most of the time biologists have been studying tardigrades, the sequence has been a mystery. For most of the time, the wrong answer was widely accepted. The wrong answer was so reasonable and so well supported by analogy with other animals that it took several decades and a remarkable amount of careful work to overturn it. The wrong answer was sugar. Specifically, it was a particular sugar called trehalose. Trehalose is a small molecule, 2 glucose units linked together, and it has an unusual property among biological sugars. When an organism dries out, trehalose can replace some of the structural roles that water normally plays inside cells. It can stabilize membranes, it can hold proteins in their folded shapes. It can, in sufficient concentrations, transition into a kind of glassy, immobile state that locks the cell's contents in place until water returns. This was known to be the trick that several other dehydration-tolerant organisms used. Brine shrimp, for instance, the small crustaceans whose dried eggs are sold as toys under the name sea monkeys, accumulate trehalose in massive quantities, somewhere around 20% of their dry weight, when they enter their dormant phase. Several nematodes do the same. Yeast cells, when starved, do something similar. So when biologists in the second half of the 20th century began to look seriously at tardigrade cryptobiosis, the natural assumption was that they would find the same thing: pile up the trehalose, vitrify the cytoplasm, hold the body in suspended chemistry until conditions improve. It was a clean hypothesis. It made evolutionary sense. The animals that did this were not closely related to one another, but the molecular trick was simple enough that it could plausibly have evolved several times, and the trehalose explanation seemed to do all the necessary work. For decades it was the textbook answer. Tardigrades survive desiccation by accumulating trehalose. Look in the dry ton and you would find trehalose. The ton is, in essence, a small block of sugar glass with an animal preserved inside it. The trouble began when people actually measured. In the early 2000s, a research group did the careful work of homogenizing large numbers of dried tardigrades, hundreds of thousands of individuals, ground down into a fine powder, and asking the simple analytical question: how much trehalose is actually in there? The answer in the model species Hypsibius exemplaris came back at around 1/100,000th% of the dry mass. Not 20%, the way it is in brine shrimp. Not 5%, not even 1. Roughly 0.02%. trace amounts, less by a factor of about 10,000 than what would be needed for the trehalose explanation to hold. This was a problem. It meant that the sugar story, which had been the textbook account for years, was not, by itself, the story. The water bear was doing something else. The decades of analogy with brine shrimp and nematodes had been pointing, it turned out, in a direction the actual data did not support. Tardigrades use a little trehalose. They benefit from it, but trehalose alone cannot explain how an animal survives losing 99% of its water and coming back from it. The work of finding the real answer is, in 2026, still going on. But the picture that has emerged over the last decade is, in its outline, now reasonably clear, and it is more interesting than the sugar version was. The clearest part of the picture comes from a laboratory in Wyoming, run by a molecular biologist named Thomas Boothby. Boothby's group, working through the genome of Hypsibius exemplaris, identified a family of proteins that the tardigrade produces in enormous quantities and that no other animal has anything quite like. The proteins are given the rather plain name CAHS, which stands for Cytoplasmic Abundant Heat Soluble. The full name is more useful than it sounds. The proteins live in the cytoplasm of the cell. There is a great deal of them, and unusually, you can boil them in water without destroying their function. Most proteins denature when heated, the way the white of an egg goes from clear to opaque, but CAHS proteins survive boiling unchanged. This last property is one of the things that drew attention to them in the first place. Whatever they are doing inside the cell, they are not behaving like ordinary proteins. What CAHS proteins are, in fact, is a class of molecule that biologists have only recently begun to take seriously: intrinsically disordered proteins. Most proteins, in the textbook picture, fold into a specific three-dimensional shape that determines their function. The shape is the whole point. Intrinsically disordered proteins do not. They flop around in the cytoplasm in an ever-changing tangle, and they do their work precisely by being shapeless. Until the early 2000s, biology had largely ignored them. They did not fit the model. We are now finding in animal after animal that a great deal of important cellular chemistry is run by these proteins, and that the tardigrade is unusually rich in them. When a tardigrade begins to dry, the CAHS proteins stop being shapeless. They begin to assemble. They aggregate into long fibrous structures and then into a kind of soft gel that fills the cell, holding everything in place as the water leaves. By the time the cell is fully dry, the gel has hardened. Whether the hardened state is technically a glass, a vitrified solid like obsidian or hard candy, has been argued about for several years now. Boothby's original papers proposed that it was. A more recent study from a Japanese group has questioned whether what looks like vitrification might in some cases be the artifact of trace amounts of water leaving slowly. The mechanism is, in 2026, not yet fully settled. What is reasonably clear is that something glass-like is happening and that the CAHS proteins are part of it, and that the glass-like state is what holds the dried tardigrade in its preserved condition for however long the dry period lasts. The trehalose, it turns out, is not absent. It is just present in small amounts, and it works synergistically with the CAHS proteins rather than carrying the load alone. Boothby's group showed this directly in a careful experiment. Yeast cells, which can be engineered to express tardigrade CAHS proteins, gained much greater desiccation tolerance when CAHS and trehalose were both present together than they did with either one alone. The two molecules cooperate. The water bear's solution is not a single trick, but a small ensemble: trehalose, CAHS proteins, and several other molecules whose roles are still being worked out. assembling itself into something larger than any of its parts. This matters beyond the specific case of the tardigrade because it changes how the problem of dehydration tolerance can be approached in human medicine. There are many things about modern medicine that depend on cold storage: vaccines, blood products, certain proteins used to treat clotting disorders, certain cellular therapies. All of them have to be kept refrigerated, often deeply refrigerated, between the laboratory and the patient. The cold chain that this requires is enormously expensive, and in much of the world it is unreliable or absent altogether. A protein that can dry biological materials into a stable, room-temperature, glass-like state, the way the tardigrade dries itself, would change a great deal about how medicine is delivered. The CAHS proteins are not yet doing this in pharmacies, but they are doing it in laboratories, and the gap between laboratory and pharmacy is, for at least one specific drug, beginning to close. That part of the story belongs to a different chapter, in a different building, and to a haemophilia patient who has not yet been mentioned. For now, the tardigrade in the drying moss has finished what it needs to do. The cytoplasm is solid. The metabolism is undetectable. The body, which an hour ago was walking and eating, is a small dark barrel curled in on itself, 0.5 millimetre across, indistinguishable from a speck of dust to anything looking down at it from human height. The moss above it dries the rest of the way. The afternoon ends. The wall holds its warmth for a while, and then it doesn't. The tun waits. What the tun is waiting for, in most cases, is water. The dry weeks pass, the dry months pass, sometimes the dry decades pass, and eventually there is rain again, or meltwater, or the slow capillary creep of moisture returning to the moss, and the animal unfolds. That is the ordinary case. There are, however, cases where something else arrives first. In a forest in normal weather, the radiation environment at the surface of a piece of moss is mild. Cosmic rays come down from the sky in low numbers. Trace amounts of natural radioactive isotopes, potassium-40 in the rocks, radon in the soil, small contributions from the slow decay of uranium and thorium, produce a steady background. None of it is enough to bother any living thing. The dose is, for practical purposes, nothing. But the tardigrade, by virtue of where it lives, is sometimes exposed to far more than the ordinary dose. A piece of moss baking on bare granite at high altitude takes the brunt of solar ultraviolet radiation in a way that a moss in a damp valley does not. A patch of lichen on the side of an Antarctic rock outcrop near the magnetic pole where the atmosphere lets cosmic rays pour through more easily sees roughly 10 times the cosmic radiation that the same lichen would see in the temperate zone. And on the kind of timescales the tardigrade lineage operates on, 500 million years, there have been events in Earth's history when the natural radiation flux was, at least locally, severe. Nearby supernovas, bursts of cosmic rays during periods of low solar activity, geological exposures of radioactive ore. The water bear has, in its long history, been radiation stressed often enough that the lineage has had to deal with it, and so at some point in the 20th century, biologists got curious about how the animal handles it. The first serious experiment was published in 1964. 3 French researchers, R. M. May, M. Maria and J. Guimard, working with X-ray equipment that was, by modern standards, fairly basic, took specimens of a tardigrade species then called Macrobiotus areolatus and irradiated them in 2 conditions. Some animals were active and hydrated, going about their tardigrade lives in a small drop of water. Others had been carefully dried, allowed to enter the tun state, and were sitting on the slide as small dehydrated barrels. The researchers exposed both groups to increasing doses of X-rays and recorded what dose was required to kill half the population within 24 hours. This is the standard measure in radiobiology, the dose at which half the exposed population dies, the LD50, useful precisely because it can be compared across species. For a human being, the LD50 from acute whole-body radiation sits at somewhere around 4 to 5 gray. A gray, in this context, is a measure of absorbed dose, 1 joule of radiation energy deposited per kilogram of tissue. 5 gray is enough to overwhelm the body's capacity to repair damage to the bone marrow, the gut lining, and the immune system. A person exposed to 5 gray and given no medical care is more likely to die than not within a month. At 8 or 10 gray, the death rate approaches certainty. At 20 or 30 gray, the body fails within hours. The Tardigrades in May's 1964 paper had an LD50 of somewhere between 5 and 6,000 gray. The number is worth letting sit for a moment because the comparison is not subtle. The dose that kills a human at the 50% threshold multiplied by roughly 1200 is the dose at which half a tardigrade population dies. A dose that would kill 900 human beings at once, delivered into a single half-millimetre water bear, is roughly the threshold at which the small water bear becomes statistically uncomfortable. Below that threshold, the animal mostly walks it off. The tardigrades that survived the experiment continued moving, eating, laying eggs. The eggs hatched. Their offspring were normal. This was, on its own, a striking finding. The 1964 paper made some small ripples in the community of radiobiologists who paid attention to invertebrates and was largely ignored by everyone else. But the finding contained, embedded in it, something stranger that took the field much longer to fully appreciate. The stranger thing is that the active tardigrades and the dried tardigrades, the ones in water and the ones in tun state, had roughly the same LD50. It is not obvious on first hearing why this is strange. To see why, you have to know how radiation does its damage inside a living cell. The naive picture, in which an X-ray photon flies in and breaks a strand of DNA on direct impact, is correct only some of the time. In a normal hydrated cell, the great majority of radiation damage, somewhere between 2/3 and 4/5 of it, depending on the type of radiation, does not happen by direct impact at all. It happens by an indirect route, through water. The radiation ionizes water molecules. The ionized water produces, almost instantaneously, a small swarm of unstable molecular fragments. Most importantly, the hydroxyl radical, an oxygen and hydrogen pairing missing one electron, which is one of the most chemically aggressive species in biology. These hydroxyl radicals, born in the cell's water, drift the very short distance to the nearest large molecules and tear them apart. DNA, proteins, membranes, all of them oxidized in the same chemical chain. Almost all of the damage flows through water. Water is, for ionizing radiation, the medium of harm. In a dried tardigrade, then, the indirect damage pathway should be largely shut down. There is no water to ionize. The hydroxyl radicals cannot form or form only at the trace level of the residual moisture that even a dry tun retains. A dried animal exposed to 1,000 gray of X-rays should, by every reasonable calculation, sustain only a fraction of the damage that a hydrated animal at the same dose would. The tun, in other words, ought to be hugely more radiation tolerant than the active tardigrade. But May's measurements said it was not. The two groups died at essentially the same dose. The advantage that physics predicts, a dramatic resilience boost for the dried form, was not there. There is only one way to read this result honestly. The hydrated tardigrade, walking around in its water drop with all the indirect damage pathways available to ionizing radiation, was somehow protecting its DNA almost as well as the dried tardigrade with the indirect pathways shut down. The active animal was not depending on dehydration to survive radiation. It was doing something else, something that worked in water, on the actively running, fully hydrated body that brought its tolerance up to nearly the same level as the dried state. What that something else was, the 1964 paper could not say. The tools to find it did not yet exist. 40 years later, in 2005, the Swedish biologist Ingemar Jönsson, working with a different species, Rictersius coronifer, an alpine tardigrade with a particularly tough reputation, repeated the experiment with modern equipment and modern dosimetry. He confirmed May's finding. The active and dried animals had the same dose response. He extended the work to gamma radiation and got the same result. Other researchers, on other species, did the same. The paradox held. By the late 2000s, it was a standard, published, well-established fact about water-bear biology that nobody had a mechanism for. It is not particularly common in modern biology for a basic question about an animal's physiology to sit unanswered for 40 years. It is rarer still for the question to remain open while the field accumulates more and more confirming evidence that the question is real. The radiation tolerance of the tardigrade was, for most of the second half of the 20th century, an established fact in search of an explanation. The animal was carrying somewhere inside its body a piece of molecular machinery that nobody could yet find. In a moss in some quiet corner of the world, on the same afternoon as the experiment in the Paris laboratory, a tardigrade is walking across a wet leaf, opening a soft cell with its stylets, drinking from it. It does not know that the radiation tolerance carried inside its body has, for half a century by now, been an unsolved problem in molecular biology. It does not know that anything is unsolved. It walks slowly, finds another cell, and goes on. It walks on. The cell wall opens, the contents are drawn in, the animal moves to the next leaflet. 6,000 kilometers away, in a laboratory at the University of Tokyo, a molecular biologist named Takekazu Kunieda is reading a genome. The genome belongs to a different tardigrade, Ramazotius varioornatus, a small terrestrial species that lives like most water bears on damp moss and lichen, but that has, in laboratory testing, the highest radiation tolerance of any tardigrade so far studied. Where Hypsibius exemplaris manages an LD50 in the 4,000 to 5,000 Gray range, Ramazotius tolerates dosing into the upper end of that distribution and beyond. If any tardigrade was likely to be carrying the unknown radiation protective machinery in particularly clear form, Ramazotius was the one to look at. Kunieda's group, in collaboration with the molecular biologist Takuma Hashimoto, had spent years doing the careful, expensive work of sequencing the animal's genome to a quality high enough to compare gene by gene with other organisms. By the autumn of 2016, they had it. The genome contained, as genomes always do, a great many genes that were familiar. The ordinary machinery shared across the animal kingdom, with minor tardigrade-specific tweaks. It contained the genes for the stylet apparatus, the genes for the cuticle, the genes for the basic chemistry of being a small invertebrate. It also contained, when carefully searched, several thousand genes that were unique to tardigrades, with no recognizable homologs in any other animal group. This was, in itself, not unusual. Most genomes contain orphan genes, sequences that have arisen during the evolutionary history of a particular lineage and have not yet been matched to anything in any other species. Most orphan genes turn out, on closer inspection, to do unspectacular work. The interesting question was always whether any of them did anything spectacular. Kunieda and Hashimoto's group focused on a particular orphan, a small, highly charged, intrinsically disordered protein that the tardigrade produced abundantly inside the nuclei of its cells, where the chromosomes are kept. Proteins that live in the nucleus and bind to DNA tend to be doing something important. The team named the protein Dsup, short for damage suppressor, and reported their findings in the journal Nature Communications in September 2016. The findings landed harder than the title suggested. What the group had done after identifying Dsup in the tardigrade genome was the most direct possible test of its function. They took the gene that codes for Dsup and they inserted it into the genome of a line of cultured human cells, specifically a kidney cell line known as HEK293, a workhorse of molecular biology. The human cells, now carrying the tardigrade gene, began producing the tardigrade protein. Dsup made its way into the human nuclei, found the human chromosomes, and bound to them in the same way it does inside the tardigrade. Then the team irradiated the cells. 2 cell populations were exposed to identical doses of X-rays, one population producing Dsup, the other an ordinary control. After irradiation, the team measured the amount of DNA damage in each. The number of double-strand breaks, the most dangerous form of radiation injury, was reduced in the Dsup-expressing cells by roughly 40%, not eliminated, not catastrophically reduced, but by a clear, reproducible, statistically robust 40%. A single tardigrade protein dropped into a human cell had given that human cell measurable resistance to ionizing radiation. This is the kind of experiment that, in molecular biology, tends to make people stop what they are doing and read the paper twice. Cross-species protein function is not in itself surprising. Molecular biology is built on the premise that the underlying machinery of life is shared, and proteins from one organism can often be made to work in another. What was striking about the Dsup result was the magnitude, the cleanness, and the implication. The protective machinery the tardigrade had been quietly carrying for several 100 million years was not so specialized that it required tardigrade biochemistry to function. It worked, more or less directly, in a human nucleus. The half-billion-year-old solution to a problem the lineage had faced in its early history was, in the laboratory, transferable. What followed Dsup's discovery was, predictably, several years of intense work to understand exactly how it does what it does. The picture that has come together, through papers from Hashimoto's group and from a separate group at the Stowers Institute in Kansas City, led by Carolina Chavez and Jim Kadonaga, is mechanically elegant. Dsup is, structurally, a long disordered protein with no fixed shape. It binds to nucleosomes, the protein spools around which DNA is wrapped inside the nucleus. Once bound, it does not lock the chromatin into a rigid state. That would interfere with the cell's ordinary activities like transcription and replication, which require the DNA to be accessible. Instead, it forms a soft, dynamic coating over the nucleosome surface. The coating is loose enough for cellular machinery to do its normal work, and tight enough to physically intercept hydroxyl radicals before they reach the DNA strand. The shield is not a wall. It is more like a flexible mesh, dense enough to catch what matters. This is, on its own, a remarkable thing for a protein to do. There are many DNA-binding proteins in the natural world. Very few of them function as physical radiation shields. Dsup appears to be tardigrade-specific evolved at some point in the lineage's deep history, possibly as a response to repeated dehydration cycles. When a cell dries out, even without external radiation, oxidative stress on the DNA goes up, and an animal whose lifestyle requires repeated drying and re-wetting needs a way to manage that damage. The radiation tolerance in this view is a side effect. The selection pressure was probably the desiccation cycle. The protection against external radiation came, evolutionarily speaking, for free. The story would be cleaner if it ended there. The tardigrade would have its mechanism, Dsup, a single elegant protein with a single elegant function, and the half-century-old paradox would be closed. But the story in 2026 does not end there. It does not even particularly stop. Two complications, both of them recent, have begun to reshape the picture. The first is that Dsup, transferred into human cells, is not always benign. A 2023 study from a research group at Brown University, led by the neurobiologist Justin Fallon, attempted to express Dsup in cultured rat neurons, partly with the thought that radiation-protected nerve cells could be useful in cancer therapy and possibly in long-duration spaceflight. The result was unexpected. In the rat neurons, Dsup did not protect the DNA. It promoted DNA damage. The chromatin organization was disturbed. Double-strand breaks went up. The cells began to die. The same protein that protected human kidney cells from radiation made rat brain cells worse. The biology underlying this discrepancy is still being worked out. The most likely explanation is that the architecture of chromatin in different cell types is sufficiently different that what shields a kidney cell genome behaves differently in a neuron, but the conclusion that any future therapy will draw from this is straightforward. Dsup is not a universal solution. It is a tool. Like all biological tools, it depends on the context it is used in. The second complication is more fundamental. Dsup is not, it has turned out, the only thing the tardigrade is doing. For years after the 2016 paper, Dsup carried the conceptual weight of explaining tardigrade radiation tolerance. It was the satisfying single answer to a long-standing question, and the field largely treated it that way. Then, in 2024, two more pieces of the picture arrived. One from a French team in Paris, one from a Chinese team in Henan, and the satisfying single answer became, again, several questions. In the meantime, the original animal, the Ramazotius whose genome started all of this, is still in a culture dish in Tokyo, eating algae, laying eggs. It does not know that its protein is in human cells, in rat neurons, in funded research grants. It does not know that anything has been found at all. It walks across its dish in the same slow, methodical way that water bears have walked across damp surfaces for 500 million years, and the protein that earned a Nature paper sits where it has always sat, in the nuclei of every cell, doing the quiet work of holding chromatin together against the next stress, whatever that stress turns out to be. The French team was not originally looking for a new protein. They were looking at the question of repair. In a laboratory at the Muséum National d'Histoire Naturelle in Paris, a building that has, in one form or another, been a centre of European zoology for nearly 400 years, a molecular biologist named Jean-Paul Concordet had become interested in the radiation tolerance of tardigrades from a particular angle. The Dsup story, as it had developed since 2016, was a story about prevention. The protein bound to chromatin intercepted hydroxyl radicals and physically reduced the amount of DNA damage the radiation managed to inflict. That was beyond reasonable doubt part of the picture, but it could not on its own be the whole picture. Damage prevention is rarely complete, and the doses tardigrades survive are extreme enough that even a 40% reduction in damage would still leave the cell with thousands of double-strand breaks per chromosome. Something had to be repairing what got through. Concord's group decided to attack the problem the way molecular biologists usually attack a poorly understood cellular response, by reading the transcriptome. When a cell faces a stress, it responds by changing which genes it uses. Some genes are switched off, many more are switched on. The collective shift in activity, the messenger RNA produced in the hours after the stress, is a record of what the cell has decided to make in response. Reading this record at high resolution is one of the most useful things modern molecular biology can do, because it tells you what the cell itself thinks the answer is. The team did this for Hypsibius exemplaris, the standard laboratory tardigrade, and then, importantly, for 2 other species. The choice of the 2 was deliberate. They picked Acutuncus antarcticus, a tardigrade endemic to Antarctica with its own long history of cold and dehydration tolerance. They picked Paramacrobiotus fairbanksi, a North American species from a different superfamily entirely. The 3 species had diverged from one another a long time ago, long before the dinosaurs in evolutionary terms, and they belong to different ecological worlds. The point of choosing 3 was simple. If the same gene was switched on in all 3 species in response to radiation, then whatever it was doing was likely an ancient, conserved part of the tardigrade radiation response. Lineage-specific quirks would not show up in all 3. Genuinely important machinery would. The team irradiated each species with gamma rays at a substantial dose, roughly 1,000 Gray, which is well below the LD50, but enough to provoke a strong response. And at intervals over the following hours, they extracted RNA from the animals and sequenced what was being transcribed. The results came back, and they were, in one specific way, very clean. There were many genes upregulated in all 3 species. Most of them were the usual cast of DNA repair proteins that any cell in any animal would deploy after radiation damage. The proteins that recognize broken strands, the proteins that splice them back together, the proteins that maintain the integrity of the genome through the long minutes of crisis. None of this was a surprise. What was a surprise was that, alongside the universal repair machinery, there was a single tardigrade-specific gene that came on in all 3 species with unusual force. The gene had no homologue in any non-tardigrade organism. It was not Dsup. It was not anything that had been described before, and it was upregulated dozens of times above its baseline level within hours of the radiation exposure. The team named it TDR1, which stands, with the slightly clinical economy of molecular biology, for tardigrade DNA damage response gene 1. The naming was the easy part. The harder part was figuring out what TDR1 did. The protein it produced, when expressed and purified, turned out to be, like Dsup, intrinsically disordered. Another shapeless, highly charged protein of the kind that the tardigrade lineage seems to be unusually fond of producing. But TDR1 did not behave like DSUP in cellular tests. It did not coat chromatin in a thin protective layer. Instead, when added to DNA in a test tube at high enough concentration, TDR1 caused the DNA to condense, to bunch up into compact aggregates. The DNA, in the presence of TDR1, was being gathered together rather than shielded. This was, on its own, an unusual property. But the function it suggested was elegant once you considered what a tardigrade cell actually has to deal with after a heavy radiation dose. The genome in such a cell has been broken in many places at once. There may be hundreds or thousands of double-strand breaks scattered across the chromosomes. The repair machinery is working as fast as it can, But DNA repair is not instantaneous. It takes minutes to hours, and during that interval, the broken ends of the chromosomes are loose, free to drift, free to be ligated to the wrong partners. A double-strand break repaired against the wrong piece of DNA is a translocation, a chromosomal scrambling that can be fatal to the cell, or worse, can survive and become cancerous. What TDR1 appeared to be doing was holding the broken pieces together, not repairing them, but keeping them in their correct neighborhoods so the repair machinery could find the right partners. A kind of cellular bookkeeping. The chromatin, condensed by TDR1, would not drift. The broken ends would stay near their original neighbors. The repair, when it happened, would happen accurately. The damage would be unavoidable. The consequences of the damage could be managed. Concordit's team, like Hashimoto's group before them, then ran the cross-species test. They put TDR1 into human cells and exposed those cells to bleomycin, a drug that produces DNA damage similar to ionizing radiation. The TDR1 expressing cells had measurably less surviving damage than the controls. Another tardigrade protein in another human cell line was working. The paper appeared in the journal eLife in 2024. It did not get the same level of public attention that the original Dsup paper had received, partly because the science had moved on by then and a single protein discovery was no longer surprising, partly because eLife is not the type of journal that produces the same press cycle as Nature. But within the field, the result mattered. It meant that the tardigrade was not relying on any single mechanism. The radiation response had at least two distinct molecular layers, Dsup at the front, physically blocking damage, and TDR1 behind it, organizing the response to the damage that got through. Two different proteins, both unique to tardigrades, both transferable to mammalian cells, working at different stages of the same problem. This is the kind of architecture in a biological system that strongly suggests there is more. There was more. Within a few months of the TDR1 paper, the third piece of the picture arrived, and it came from a direction that the field had not, by and large, been looking. It came from China, from a mountain in Henan province, from a sample of moss collected 6 years earlier by a local research team. The story of that sample and of what was found inside the genome of the small water bear that was crawling on it is the story that resets, more than any other piece of recent tardigrade biology, the question of what an animal is actually allowed to do. What an animal is allowed to do in the most general sense is what its genome lets it do. The genome is the inheritance, the long molecular memory of every solution the lineage has ever stumbled into and kept. An animal is, in a strict sense, the living expression of its own ancestry. It can do what its ancestors could do, modified by whatever mutations have arrived since. It can build the proteins its ancestors built. It can run the chemistry its ancestors ran. The boundary of what it can do is, in normal circumstances, the boundary of what its lineage has ever done. This is not, it turns out, the whole story. In the spring of 2018, in the Funyu mountain range in Henan province, in the center of China, a research team from Qingdao Agricultural University was collecting moss. The mountains there are forested, humid, full of the kind of damp microhabitats that water bears favor. And the team, led by a young biologist named Lei Li, had been systematically surveying the local tardigrade fauna for several years. On one particular collection trip, in a moss sample taken from a mid-elevation slope, the team found something they did not recognize. The animal was a tardigrade, clearly, but it did not match any of the species described from the region. The claws were slightly different. The cuticle ornamentation was unusual. After the careful taxonomic work that confirming a new species requires, the animal was named Hypsibius henanensis, the Hypsibius from Henan, and the team began to study it. They sequenced its genome. They worked slowly through the catalogue of its proteins. They subjected it to the standard battery of stress tests, including exposure to heavy ion radiation at extremely high doses. The animal handled the radiation about as well as the most resistant tardigrades on record. The team began to look in detail at which genes were responsible. The work took 6 years. The paper that emerged from it appeared in the journal Science in October 2024, and it described 3 separate molecular contributions to the radiation tolerance of Hypsibius henanensis. 2 of these were tardigrade-specific in the now familiar way. There was a disordered protein, broadly comparable to TDR1 in its function, that the team called TDP1. There was a mitochondrial modification that boosted the cell's production of NAD+, a small molecule that DNA repair enzymes need to do their work. Both were interesting. Both extended the picture that Concordet's group had begun to assemble. Neither was the part of the paper that, when biologists read it, made them stop reading and start over. The part that made them stop reading was the third mechanism. In the genome of Hypsibius henanensis, the Chinese team had found a gene that should not, by any reasonable account, be in an animal at all. The gene was a recognizable functional copy of an enzyme called dopa dioxygenase, often abbreviated DODA1. DODA1 catalyses a specific chemical reaction in the metabolic pathway that produces a class of pigments called betalains. Betalains are the molecules that make beetroot deep red, that make dragonfruit pink, that give certain mushrooms and cactus flowers their saturated colors. They are, in nature, produced by a small range of plants, by some fungi, and by certain bacteria. They are not anywhere in the conventional taxonomy of biology produced by animals. The genes for making betalains do not exist in animal genomes because animals diverged from plants and fungi billions of years ago, and the betalain pathway evolved on the other side of that divergence, except in this tardigrade. The DODA1 gene in Hypsibius henanensis is not similar to any other animal gene. It is similar to a bacterial gene. The closest matches in the global sequence databases come from soil bacteria, the ordinary kinds of microbes that live in damp earth, in moss, in the films of moisture that tardigrades themselves spend their lives in. The most parsimonious explanation, given the sequence evidence, is that at some point in the deep history of the tardigrade lineage, possibly 500 million years ago, possibly more recently, a bacterium inside or around an early tardigrade transferred a copy of this gene into the tardigrade's germline DNA. The bacterium itself did not survive in any continuous sense. The gene did. It was passed down, generation after generation, embedded in the chromosomes of every descendant water bear, until it ended up in Hypsibius henanensis in 2018 in Moss in central China, where a research team eventually noticed it. This kind of event has a name. It is called horizontal gene transfer. The conventional picture of inheritance is vertical. Parents pass genes to offspring. The lineage moves forward in time. The genealogical tree branches and never reconnects. Horizontal transfer is when genes move sideways between organisms that are not parent and child, sometimes between organisms in entirely different kingdoms of life. It is now known to be common in bacteria, where it is one of the main reasons antibiotic resistance spreads so quickly through the microbial world. It is far rarer in animals, but not absent. The tardigrade lineage in particular appears to have collected a small but significant set of genes from non-animal sources over its 500 million year history. Some are bacterial, some are fungal, a few are from plants. The water bear's genome contains, in fragments, a record of intimate exchanges with a great many things that are not other water bears. What makes the DODA1 case so remarkable is not just the borrowing. It is the use the tardigrade has put it to. The Chinese team showed, in a series of careful biochemical experiments, that Hypsibius henanensis uses its borrowed DODA1 to produce betalains in its own body. The pigments are made on demand, not constantly. When the animal is exposed to radiation, or by extension to other forms of oxidative stress, the DODA1 pathway turns on, the betalains are synthesized, and the animal's tissues fill with the same family of antioxidant molecules that a beetroot uses to protect itself from sunlight stress. The betalains are extraordinarily good at neutralizing the reactive oxygen species that radiation generates. They mop up the chemical fragments before those fragments reach the DNA. They do, in chemical terms, what plants do. A small animal defending itself against ionizing radiation by deploying a piece of plant pigment chemistry encoded by a gene borrowed from a bacterium 500 million years ago. This is not a sentence that biology, as it has been taught for the last 150 years, comfortably accommodates. The kingdoms, animals, plants, fungi, bacteria, were drawn as separate domains, with their own characteristic chemistries, their own metabolic strategies, their own evolutionary trajectories. The drawing was useful. It is also, in the actual living world, a simplification that obscures something the data is now making increasingly clear. Genes move. Chemistry crosses lineages. The boundaries between the kingdoms, while real in many ways, are also porous in ways that the textbook diagrams do not show. The tardigrade in central China is, in the most literal possible sense, an animal that contains a working piece of bacterial inheritance that produces plant chemistry. It is not a metaphor. It is not a curiosity. It is a measurable, reproducible, experimentally verified fact about an animal that lives in moss. The Chinese team did not stop with the demonstration in the tardigrade. They synthesized the betalains the tardigrade produces. They added them to cultures of human cells. They irradiated those cells. The cells with the tardigrade's betalains survived radiation that would have killed a far larger fraction of the controls. The plant chemistry, encoded by the bacterial gene, harbored for 500 million years inside an animal lineage, also worked in human tissue. There is a point when reading a result like this where it is worth pausing. The animal in the moss in Henan is not aware that any of this has happened. It does not know that its DNA contains an inheritance from a bacterium that died, in the most literal sense, in some Cambrian or pre-Cambrian moment that no one will ever recover. It does not know that the pigment it produces under stress is the same chemistry that colors the foods on a human dinner table. It does not know that the pigment, extracted from its body and given to human cells, can protect those cells from radiation. The animal does what it has always done. It walks across damp moss. It opens cells with its stylets. When the world becomes hostile, it folds. When the world returns, it walks again. What is sitting inside its body is older than almost everything in the visible biology of Earth. The bacterium that gave it the gene is not even a memory anymore. There is no remaining lineage, no name, no fossil, nothing except the sequence preserved in the tardigrade's chromosomes. The plants that use the same chemistry today evolved their version of the pathway entirely separately on their side of a kingdom-level divide, hundreds of millions of years after the tardigrade had quietly been carrying its borrowed copy. The fact that the chemistries match is, in a real sense, an accident of evolutionary convergence around what works against oxidative damage. Or perhaps not an accident, perhaps a clue, Perhaps the constraints on biological chemistry, what kinds of molecules can usefully neutralize free radicals, what kinds of pathways can be cobbled together from common precursors, are tight enough that there are only a few good answers, and life, wherever it has run into the problem, has converged on the same handful of solutions. The tardigrade and the beetroot and the soil bacterium are, in this view, all using something close to the only chemistry that works. The differences between them are mostly cosmetic. Either way, the picture has changed. The water bear's radiation tolerance is not one trick or two tricks. It is at least 4 tricks, each with its own mechanism, its own evolutionary history, its own degree of strangeness. There is Dsup, the chromatin shield evolved within the tardigrade lineage, possibly as a desiccation adaptation that incidentally handles radiation. There is TDR1, the chromatin organizer, holding broken DNA in place while repair happens. There is TDP1, the disordered repair assistant in Hypsibius henanensis. And there is DODA1, the borrowed bacterial gene producing plant chemistry to clean up oxidative stress. Each of these does a different part of the job. Together they explain the half-century-old paradox that May noticed in 1964, the fact that hydrated and dehydrated tardigrades have the same radiation tolerance, because the protection is not coming from dehydration. It is coming from the layered molecular architecture the lineage has built up over 500 million years to handle a kind of damage that the lineage has, repeatedly, had to handle. The animal in the Henan moss continues to walk. Somewhere on a research bench in Qingdao, a culture of Hypsibius henanensis is being prepared for the next experiment. Somewhere in a lab in Iowa, the same animal's relative, Ramazotius varieornatus, is contributing its Dsup gene to a clinical trial that has not yet formally begun. Somewhere on a mountain in central China, the moss is wet again after a morning rain. The water bears are walking. They have, between them, more solutions to the problem of staying alive than most lineages have ever needed. They have also, between them, almost no idea that any of this is happening, which is, in some ways, the most generous thing that can be said about an animal. The animal has, for most of its evolutionary history, been entirely unaware of the sky. It lives at the scale of moss leaflets and water films. The whole vertical extent of its world is rarely more than a few centimeters, the depth of the moss patch, the height of a piece of lichen on a rock face, the thin habitable layer between the surface of the soil and the air that flows above it. The sun is, for the tardigrade, mostly an inconvenience. It dries the moss out. The moon is irrelevant. The stars are a thing the lineage has never had the equipment to perceive. The water bear has, in the most literal way, no relationship with the cosmos. It has had, until very recently, no occasion to develop one. In September 2007, a small number of tardigrades, about 3,000 of them, divided between 2 species, were prepared for the first occasion in the lineage's history. The preparation happened in a laboratory at Kristianstad University in southern Sweden, under the direction of an evolutionary biologist named Ingemar Jönsson. Jönsson had spent years working on tardigrade desiccation tolerance, and he was one of the small number of researchers who, by the mid-2000s, had begun to think seriously about whether the same physiology that protected the animal from drying might also protect it from the conditions of outer space. The European Space Agency, planning a series of small biological experiments aboard a recoverable orbital satellite, agreed to give him the chance to find out. The mission was called Photon M3. The tardigrade payload, given the kind of acronym that scientists give to projects when they cannot quite resist, was named TARDIS, Tardigrades in Space. 2 species were chosen, Rictursius coronifer, an alpine species with a long history of laboratory work behind it, and Milnesium tardigradum, a slightly larger predatory tardigrade common in temperate moss. Both species were known to enter robust ton states when dried, and both had reasonable radiation tolerance in laboratory tests. The animals were collected, dehydrated, and arranged in small holders that would expose them to one of three conditions during the flight: vacuum alone, vacuum plus the full ultraviolet A range of solar radiation, or vacuum plus the unfiltered ultraviolet B range that hits anything in low Earth orbit without an atmosphere to absorb it. On the 14th of September 2007, the Photon M3 capsule lifted off from the Baikonur Cosmodrome in Kazakhstan on a Soyuz rocket, carrying its mixed scientific payload into orbit at an altitude of approximately 270 kilometers. The tardigrades were among the cargo. They were, at the moment of launch, dehydrated tons in steel exposure trays. They were, in any biologically meaningful sense, not awake. What happened over the next 12 days has no real precedent in the history of biology. When the capsule reached its operating orbit, an external panel called BioPan opened and exposed the experimental trays directly to the space environment. The pressure surrounding the tardigrades dropped to the standard hard vacuum of low Earth orbit, somewhere around 10 to the -6 millibars, which is the pressure of effectively nothing. Half the gases that constitute Earth's atmosphere were not present. The temperature, depending on whether the trays were in sunlight or in shadow, swung between roughly -30 and +60 degrees Celsius. Cosmic rays, ordinarily filtered out by the atmosphere, struck the trays at the full intensity that interplanetary space delivers to anything in orbit. For the ultraviolet exposed groups, the unfiltered solar UV, including the high-energy UVB and UVC wavelengths that the ozone layer absorbs before they can reach the surface, fell directly onto the dehydrated bodies. These are not survivable conditions for any organism that has ever been tested. The combination of vacuum, temperature swing, cosmic radiation, and unfiltered solar UV is, in principle, a category of environment that lies outside the operating range of life. Until this experiment, every multicellular animal that had ever been exposed to it had died very quickly and unambiguously. The capsule re-entered Earth's atmosphere on the 26th of September and was recovered from a field in Kazakhstan. The BioPan trays were brought back to Sweden. Jönsson's team rehydrated the tardigrades. Most of the animals from the vacuum-only groups, both species, walked. This was the result that, when Jönsson and his colleagues published it in Current Biology in 2008, made the international press for several weeks. Tardigrades had become, formally, the first animals in the history of multicellular life to survive direct exposure to open space and return to active biology afterwards. The vacuum by itself had not killed them. The cosmic radiation by itself had not killed them. They had walked off the BioPan tray after 12 days in conditions that no other animal had ever endured, and several of them, when given food and water, had gone on to lay normal eggs that hatched into normal offspring. The vacuum plus UV groups did not fare as well. The UVA group had reduced survival but produced some live animals. The UVB group, with its higher energy radiation, had only a few survivors. UV, it turned out, was the difficulty. Without an atmosphere to filter it, ultraviolet light at the wavelengths that reach low Earth orbit damages biological molecules, DNA, lipids, the cuticle proteins of the tardigrade itself, at a rate that even the lineage's accumulated repair machinery cannot fully manage in 12 days. Vacuum, the famous lethal condition, was not the lethal condition. Light was. The animal had spent 500 million years quietly developing radiation defenses and dehydration tolerance and the molecular architecture of suspended life, and what those defenses turned out to handle most easily was the part of space that everyone had assumed would be hardest. The part everyone had assumed would be straightforward, sunlight without atmospheric filtering, was the part that mattered. It is easy now to underestimate how strange the TARDIS result was when it landed in 2008. Until that moment, the question of whether any animal could survive open space had been entirely theoretical. Spores and bacteria, certainly. There was good evidence that some hardy microbes could weather brief exposures. Dried seeds, possibly. Animals, no. The combination of physiological complexity and the scale of the body, even a tardigrade, microscopic by ordinary standards, is enormous compared to a bacterial spore, was supposed to make the project of surviving vacuum, cosmic rays, and temperature extremes simultaneously a non-starter. Most biologists, asked beforehand, would have predicted that the tardigrades would come back dead. The fact that they came back alive in numbers was a quietly seismic event. It meant that the ceiling on what an animal body could endure had been, in a single experiment, raised dramatically. The implication that received the most attention was, predictably, the speculative one. If a tardigrade can survive 12 days in open space, then perhaps an animal of this kind, embedded in a rock blasted off a planet by an asteroid impact, could in principle survive interplanetary travel. The lineage might, in theory, ride a meteorite from one world to another, the old hypothesis of panspermia brought back into modern conversation by an animal small enough to fit on the head of a pin. Whether this has ever actually happened anywhere in the solar system is unknown and probably unknowable. What the TARDIS experiment established was only that it is not, on biological grounds, impossible. The more interesting implication in the actual practice of biology was about Earth. The lineage had developed all of this without any selective pressure from space. There were no ancestral water bears that needed to handle vacuum or cosmic rays in the ordinary course of their lives. The capacity to do so was a side effect, a bonus property of the dehydration response, the radiation tolerance, and the various molecular layers the animal had built up to handle the much more mundane stresses of moss-dwelling life. Drying out, freezing, ultraviolet damage from intense sun on exposed mountain habitats, the occasional radioactive geological substrate, these were ordinary problems for tardigrades. Open space was the same problems, scaled up, applied simultaneously, with no biological equivalent in the lineage's evolutionary history, and the equipment for handling ordinary problems turned out at the limit to handle extraordinary ones. Jönsson published a follow-up study in 2016, 8 years after the original, looking at whether the offspring of the space-exposed tardigrades showed any inherited damage. The answer was that they did not. Whatever DNA injury the parent generation had accumulated during their 12 days in vacuum had been either repaired in full or not transmitted to the next generation. The line continued. The descendants of the space tardigrades by 2016 were several generations removed from their ancestors' brief tour of low Earth orbit, and were behaving like ordinary water bears, in ordinary moss, in ordinary laboratories. There is a small, slightly absurd detail about the TARDIS experiment that is worth mentioning. The tardigrades, throughout their 12 days in space, were entirely unconscious. They were dried tons. The vacuum did not bother them because their physiology, in the ton state, does not require atmospheric pressure to maintain integrity. The radiation arrived, and the molecular shields and repair mechanisms ground through it. The temperature swung, and the animal's lack of metabolism meant they were not subjected to the thermal stress an active body would have suffered. From the tardigrade's perspective, which is a strange phrase to use, because there is no perspective in a ton, no awareness, no time, nothing happened. The animals went into the trays in Kazakhstan. They came out of the trays in Kazakhstan, and the intervening 12 days were a single seamless interval that did not include the journey to orbit, the unfiltered cosmos, or the return. The first animals to survive open space did not know they had been there. This is, in its own quiet way, one of the most accurate descriptions of tardigrade biology that has ever been written. The animal does what it does because the lineage has, over an enormous span of time, built the equipment to do it. The equipment runs whether or not the animal is paying attention. The animal is, in the deepest sense, never paying attention. Its survival is automatic, embedded in chemistry, requiring no decision, no awareness, no engagement with the conditions it is enduring. A water bear in a moss patch in a Swedish garden is running the same internal machinery as a water bear in a tray in low Earth orbit. The difference between the two situations is a difference in the surroundings, not in what the animal is doing. The animal in both cases is folded into itself, waiting. In Kazakhstan that September, 3,000 small bodies came down through the atmosphere inside a capsule that had carried them through 12 days no animal had ever been through before. They settled to Earth without ever having known they had left. There is a temperature in physics below which it is not possible to go. Absolute zero is the bottom of the thermodynamic ladder, the point at which the random thermal motion that defines temperature simply stops, where atoms hold themselves in their lowest possible energy states and have nothing further they can give up. It corresponds to minus 273.15 degrees Celsius, give or take. Nothing in the universe naturally reaches it. The cosmic microwave background, the residual radiation left over from the early universe, holds the rest of empty space at about 2.7 degrees above absolute zero, which is as cold as anywhere in nature ever gets. To go colder than that, you have to build the apparatus yourself in a laboratory, using a small chain of refrigeration techniques that work down through fractions of a degree until you arrive in territory that no part of the natural world has ever occupied. In December 2021, in a laboratory at the National University of Singapore, a small team of physicists put a tardigrade somewhere very close to that bottom. The tardigrade was a Ramazoteus varionatus, the same species whose genome had given the world the Dsup protein 5 years earlier. The animal had been collected from a roof gutter in Denmark, brought to the lab, dried into the ton state, and placed inside a piece of equipment called a dilution refrigerator, a device used in low-temperature physics to cool small samples to within a tiny fraction of absolute zero. The ton was positioned between two superconducting circuit elements called transmon qubits, a kind of artificial atom used as the basic unit in many quantum computers. The whole assembly was then cooled. The temperature reached 10 millikelvin, which is to say 1/100th of a degree above the absolute floor of thermodynamics. The pressure was reduced to roughly 6 times 10 to the -6 millibar, a vacuum considerably harder than the one the TARDIS tardigrades had endured in low Earth orbit. And there the small dehydrated body sat between two qubits in a quantum experimental apparatus for 420 hours. The reason the experiment was done has to do with quantum entanglement. The team, led by the Singaporean physicist Kwan Cheng Lee, with collaborators in Denmark, Poland and the United Kingdom, wanted to see whether a complex multicellular organism could be coupled to a quantum bit in such a way that the animal and the qubit shared a single quantum state. They reported that it could. They wrote up their findings, posted them to the preprint server Archive shortly before Christmas of 2021, and the resulting story, predictably, found its way into the popular science press in the form of a headline that more or less wrote itself. Scientists entangle a tardigrade with a qubit. First quantum entangled animal in history. The headline did not survive contact with the physics community. Within days, working physicists were publicly questioning whether the experiment had demonstrated quantum entanglement in any meaningful sense. The criticisms were technical, but they shared a common shape. The argument from people like the science writer and physicist Ben Brubaker and the Caltech-trained quantum engineer Clarice Aiello was that placing a small dielectric object, which is what a frozen tardigrade essentially is, next to a superconducting qubit shifts the qubit's resonant frequency through ordinary electromagnetic effects that have been understood for over a century. A grain of dust placed in the same position would do something similar. The shift the team observed was real. It was just not, in the technical sense, evidence of quantum entanglement, because the tardigrade was not coupled to the qubit in a way that required quantum mechanics to describe. It was coupled in a way that classical physics handles perfectly well, and labeling that coupling entanglement was, the critics argued, an overreach. Most physicists, by 2026, regard the question as essentially settled. The experiment did not entangle a tardigrade with a qubit in the way that physicists use the word. The paper, 4 years on, has not been published in a peer-reviewed journal. It remains in its preprint form. One of the original authors, the Oxford physicist Vlatko Vedral, posted a response to the criticisms in early 2022 attempting to defend the quantum interpretation, but the response did not move the consensus. The popular framing, first quantum entangled animal, has, like many such framings, outlasted the science it was originally based on. What is true about the experiment, beyond reasonable doubt, is the part the headline got wrong by leaving it small. A multicellular animal with 8 legs and a digestive tract and a nervous system was held for 17.5 days at 1/100th of a degree above absolute zero, in a vacuum harder than orbital space, and then warmed back up. When it was rehydrated, it walked. The lineage's tolerance for extreme conditions on this experiment alone was extended several orders of magnitude past what the TARDIS mission had established. The animal had visited a region of physical space that no biological organism on Earth had ever previously occupied, a region colder than anywhere in nature, a region whose existence depends on human laboratory technology, and it had returned to ordinary water-bear life on the other side. Whether the experiment was good quantum physics is a question for physicists. That the animal did the thermodynamic part of it is a fact, and the fact is, on its own, enough. The quantum framing is the kind of thing that makes science journalism interesting and frustrating in roughly equal measure. The actual result was already extraordinary. Adding a wrong descriptor on top of it did not make the result more remarkable, it only made the descriptor more visible. The thermodynamic record stands. The tardigrade still went where no animal had gone and came back. The question of what was or was not entangled with what is, for biology, almost beside the point. While that experiment was being written up and disputed, an older story was developing in a place much harder to get to. On the 11th of April 2019, a small Israeli spacecraft called Bereshit, the Hebrew word for "in the beginning," the opening word of Genesis, was attempting to land on the moon. The mission was a private one, run by an Israeli non-profit called Space IL, together with Israel Aerospace Industries, and Bereshit was small as lunar landers go, a little under 2 meters across, weighing about 150 kilograms after fuel was burnt off. Its target was a region in the northern hemisphere of the lunar near side called Mare Serenitatis, the Sea of Serenity. The descent went well until the final minutes, when one of the spacecraft's gyroscopes failed. The main engine cut out shortly after. Bereshit struck the lunar surface at high speed and was destroyed. What did not entirely get destroyed was its cargo. Among the items Bereshit was carrying, alongside its scientific instruments, an Israeli flag, copies of the Torah and the Israeli Declaration of Independence, drawings made by Israeli children, and a set of digital archives prepared by an American non-profit called the Arch Mission Foundation, was a small embedded library encoded across 25 thin layers of nickel. The library had been designed to last millions of years on the lunar surface as a kind of long-term backup of human civilization. It contained, among many other things, almost the full English Wikipedia, several thousand classical texts, and language primers intended to allow some future intelligence to reconstruct what the contents meant. Encased in the resin between the upper layers, without the prior knowledge of NASA, the European Space Agency, or any of the international bodies that ordinarily oversee planetary protection, the Arch Mission Foundation had embedded several thousand dehydrated tardigrades. The reasoning, by the Foundation's own account, was symbolic and biological at once. The library was meant to preserve the data of life on Earth, and tardigrades, small, durable, capable of surviving extended desiccation, were a way of preserving an actual sample of terrestrial life alongside the information about it. The intent was that, in some imaginable future, the tardigrades and their accompanying records might be retrieved, rehydrated, and revived. The decision to include them was made privately. There was no formal review. The first many planetary protection officials heard about it was after the spacecraft was already on its way. When Bereshit crashed, the question of what had happened to the embedded organisms became, briefly, an item of public interest. Lunar reconnaissance orbiter imagery taken in the weeks following the impact showed a debris field consistent with high-speed disintegration, but the central wreckage appeared substantially intact. A study published in 2021 by the planetary scientist Alejandra Traspas, then at Queen Mary University of London, used impact mechanics to estimate the survival probability of the tardigrades during the crash. The conclusion was that they had almost certainly been killed by the impact itself. The deceleration forces, even on small dehydrated bodies embedded in epoxy, were too high for tardigrade physiology to survive. Whether any individuals slipped through the gradient of forces and remained viable is, in Traspas's analysis, vanishingly unlikely. What remains undisputed is that the bodies are still there. Whether they were killed on impact or survived for some moments afterward, the dehydrated tons of several thousand tardigrades are now distributed across a small region of Mars Serenitatis, embedded in fragments of nickel and resin and the structural debris of the lander. They cannot revive themselves. There is no water on the surface of the Moon. There is, on the lunar near side, no atmosphere to slow any future temperature swing or shield against the unfiltered solar UV. The bodies are subject to a slow cosmic ray bombardment that will, over the course of geological time, gradually reduce them to molecular fragments. They are, in the technical sense, neither alive nor in the cryptobiotic state that allows for revival. They are something stranger, biological remnants on a world that has never had biology, sitting in conditions that resemble the dehydrated toon state in some respects and contradict it in others. If a future mission were ever to retrieve them, and if the retrieval somehow happened to preserve the chemistry of even one specimen well enough that rehydration could be attempted, no one knows whether anything would happen. The sample size for testing this question is 0. The animals on the Moon may, in principle, be biologically recoverable. They may also, in practice, be permanently inert. The honest answer is that the question has not been tested and may never be testable, because returning specifically to the Bereshit impact site to collect tardigrade bodies is not a mission anyone is currently planning. There is, in this whole arrangement, an unusual situation that the tardigrade has produced almost by accident. Several thousand specimens of an Earth lineage, about 500 million years older than the human one, are presently sitting on another world in a state of suspended biology with no immediate way home. The lineage has, in this small particular sense, become extraplanetary. It did not seek the situation out. The decision was made by a non-profit foundation in California, and the rocket was Israeli. And the whole business has the slightly absurd quality that creeps into anything happening at the intersection of private space ventures and uncoordinated planetary protection. But the bodies are there. They have been there for nearly 7 years. They will, in some distributed, weathered, slowly disintegrating form, be there long after most of the buildings on the Earth-facing side of the Moon's lunar gaze are gone, because there is nothing on the Moon to disturb them. A small animal that has spent 500 million years walking through damp moss is now also on the Moon. It does not know. The water bear has, again, no relationship with the cosmos. It is in space accidentally, and whether on a research bench in Singapore at temperatures lower than any star or in a crater on a body that humans have stood on, the relationship runs only one way. The cosmos has acquired a relationship with the tardigrade. The tardigrade has not noticed. In the late southern spring of 1983, in a valley on the eastern edge of Antarctica called Yukidori, a researcher with the 24th Japanese Antarctic Research Expedition collected a sample of moss. The valley sits on the Sawyer coast of Dronning Maud Land, at 69 degrees south, on a part of the continent that is bare rock for much of the brief summer and frozen solid for everything else. Yukidori means snowbird in Japanese, named for the snow petrels that nest on the cliffs above. The vegetation is sparse. Almost everything alive on that part of Antarctica is microscopic, but in summer the meltwater encourages thin patches of moss to grow on the protected sides of stones, and it is in these patches that most of Antarctica's terrestrial animal life makes its home. The researcher collected a piece of Bryum argenteum, a small silvery moss common in cold environments, and labeled it with a catalogue number. The sample, designated F01096, was packed for return to Japan along with the rest of that summer's collections. In Tokyo, at the National Institute of Polar Research, the moss was placed in long-term storage at -20 degrees Celsius. The freezer it went into was not a particularly remarkable freezer. It was the kind used for biological samples in any reasonably equipped research collection. Cold enough to suspend almost all chemistry, accessible enough to retrieve samples for study when needed, ordinary in every visible way. The piece of moss sat in this freezer in its labelled container, untouched, for 30 years and 5 months. The world outside the freezer changed. The Cold War ended. The internet became a thing people used in their homes. Several of the researchers who had been on the original expedition retired, and a few of them died. The institute itself was renovated more than once. The moss did not change. The animals living in it did not change either, although they had stopped being, in any conventional sense, alive. In May 2014, a polar biologist named Megumi Tsujimoto opened the container. Tsujimoto had been studying Antarctic tardigrade biology for several years, and she had become interested in a question that had been hovering at the edges of the field for a long time. The known limit in 2014 for tardigrade survival in long-term cryptobiosis was about 9 years for dehydrated tons and somewhat less for frozen specimens. There were folklore claims of much longer revivals. A famous 19th century paper had reported reviving moss tardigrades from a sample over 100 years old, but the methodology was not, by modern standards, trustworthy enough to count. The current honest record was 9 years. Tsujimoto's question was whether anything older might survive if the conditions of storage had been favorable. And what better candidate than a piece of frozen Antarctic moss that had been sitting at -20 degrees undisturbed for 3 decades? She defrosted the sample slowly at 3 degrees over 24 hours. She added water gently and let it soak for another 24. Under a stereo microscope, she examined the rehydrated moss. She found, embedded in it, the bodies of several tardigrades and a small number of eggs. With a pipette, she transferred 2 adult animals and 1 egg onto fresh agar plates, supplied them with the green algae they would need to feed on if they revived, and waited. The first day, one of the 2 adults, the one she labeled SB1, moved the rearmost pair of its 8 legs, just slightly. That was all. The other adult, SB2, did not move. The egg, SB3, was unchanged. For the next 2 weeks, Sugimoto watched. SB2 did not recover. The animal had, in some way her observation could not resolve, sustained too much damage during its 3 decades in the freezer, or perhaps had been already weakened when collected, and it did not return. SB1, however, slowly did. The leg movements increased. By the end of the first week, the animal was managing small twitches of its other legs. By the 10th day, it was attempting, in a halting and uncertain way, to walk. By the 14th day, it was walking with reasonable competence across the agar surface and beginning to feed on the algae. The recovery had taken twice as long as a normal tardigrade revival from a fresh toon. Whatever damage had accumulated over 30 years had taken time to repair, but the animal was repairing it. SB3, the egg, hatched on the same timeline. The juvenile that emerged was a normal Acutuncus antarcticus, the species endemic to Antarctica, and it began its life in 2014 as the offspring of parents that had been alive in 1983. There was no difference in any examination Sugimoto could perform between the new juvenile and any other juvenile of its species hatched from eggs only weeks old. SB1, when she had recovered enough to feed regularly, began to lay eggs, 19 of them, 14 hatched. The hatchlings were normal in every observable way, and they grew, and they laid eggs of their own. The line continued. The paper Sugimoto and her colleagues published in Cryobiology in early 2016 was modest in its claims. It described carefully what had been done and what had been observed. It established a new minimum record for tardigrade cryptobiotic survival, at least 30 years and 5 months, and it noted that the recovery process had been substantially slower than for short-term revivals, suggesting that some kind of cumulative repair work was happening during the recovery interval. It did not claim that 30 years was the limit. It claimed only that 30 years had been observed in this one case, in these specific conditions, and that the animal had walked out of it. There is something worth holding still and looking at in the small detail of this story. For 3 decades, while the world above the freezer was doing everything it does, this animal was doing nothing. It was not asleep in any way that the word usually means. Sleep involves an active nervous system, ongoing metabolism, a body engaged with the passage of time. SB1 was not engaged with time. The chemistry that defines being alive had been reduced to something so close to zero that for many years biologists could not detect any of it at all. The animal's cells were intact, the DNA was largely whole, the protein architecture was held in place by molecular machinery, the lineage had refined over 500 million years. But the work of being a tardigrade, the moment-by-moment chemistry of feeding and moving and reproducing, had stopped. SB1 in 1983 did not, in any meaningful sense, experience the next 30 years. The next thing she experienced, after the moss began to dry on the cold rock in Yukidori, was Sugimoto's pipette in 2014. It is the kind of detail that, examined directly, does not fit cleanly into ordinary categories. SB1 was not dead during those years. Death is, by every working definition biologists use, not reversible. SB1 had not gone anywhere death goes. She had also not, during those 3 decades, been alive in the active, metabolic, time-experiencing sense that the word usually carries. She had been in a third state, the state the lineage developed for situations exactly like this. Situations where the world has stopped being survivable and the animal has decided, through whatever the molecular equivalent of decision is in a creature with 200 neurons, to wait. Waiting, in this case, lasted long enough for an entire human life arc to begin and continue. A child born in 1983 who happened to encounter SB1's revival in 2014 would have been 30 years old by then. Most of the people who collected the original sample were no longer at the Institute. The Antarctic expedition that produced the sample had become a historical footnote in the Institute's records. The Yukodori Valley had been visited by other expeditions in the intervening years, and other moss had been collected from it, and a generation of Antarctic biology had come and gone. SB1 was, throughout, in the same state, folded, dry frozen, indistinguishable from a speck of debris on a slide. There is no perfect way to describe what kind of thing this is. The ton is not a coma. Comas have ongoing biology. The ton is not death. Death is, by definition, terminal. The ton is its own category, the lineage's particular invention, an evolutionary solution to the problem of inhabiting a planet that is intermittently uninhabitable. What it does, more than anything else, is decouple the animal from time. The tardigrade in the ton is not living through the dry years. It is, in a way, that the language of biology has not yet developed clean words for skipping them. SB1 lived for several months after her revival. She laid her 19 eggs, watched them hatch, fed normally, behaved like an ordinary Acutuncus antarcticus, and died of natural causes some weeks after the last hatching. Her descendants, by 2026, are several generations into the laboratory. They are still being maintained at the National Institute of Polar Research in Tokyo, on agar plates with green algae, the way SB1 was kept after her revival. The line, in a continuous biological sense, runs from the moss in Yukodori in 1983 through one paused individual through her offspring to the present. There is no break in the chain except the gap of 3 decades when the chain was not, technically, doing anything. It is not a particularly large fact in the wider scheme of tardigrade biology. The DODA1 result is more important. The clinical applications are more consequential. The space experiments have wider cultural reach. SB1 is, in the bibliography of her field, a single entry in cryobiology with a moderate citation count. But she is also a small, specific named creature who, in 1983, on a piece of Antarctic moss, decided to stop, and in 2014, on a piece of agar in Tokyo, decided to start again. The interval between those 2 decisions was 30 years and 5 months and contained, among other things, the entire research career of several of the people who would eventually study her. The animal does not know any of this. Then again, neither in any deep way do we. The thing she did between 1983 and 2014 is, in the most honest sense, something the biology of larger creatures has no equipment for. We can describe it, we cannot do it. A drop of water, in May 2014, fell onto a small dry body in a Tokyo lab. The animal that had been waiting very precisely since the autumn of 1983 began to move. The animal began to move. By the time SB1 was walking across her agar plate in Tokyo in 2014, a quieter version of the same story was already underway in laboratories thousands of kilometers apart, where pieces of tardigrade biology were being gradually extracted, purified, and put to work inside human cells. The haemophilia clinic in question is, for the purposes of this story, hypothetical, but the situation it describes is not. There is a class of medication called factor VIII, which is, in chemical terms, a complicated protein that the human bloodstream uses to make blood clot. People with haemophilia A genetic condition that affects roughly 1 in 5,000 males worldwide, do not produce enough functional factor VIII on their own. They bleed, sometimes catastrophically, from injuries that would be ordinary for everyone else. The standard treatment is to inject the missing protein, manufactured pharmaceutically, on a regular schedule. The trouble with factor VIII is that it is, like most therapeutic proteins, deeply unstable at room temperature. The complicated three-dimensional shape that allows it to do its work in the bloodstream falls apart when the molecule is warm, dehydrated, or roughly handled. To remain useful, factor VIII has to be kept refrigerated and ideally cold-chained from the manufacturer to the pharmacy to the patient. In the wealthy parts of the world, this works reasonably well. In rural sub-Saharan Africa, in disaster zones after earthquakes or floods, in conflict regions where infrastructure has collapsed, in remote clinics that depend on intermittent electricity, the cold chain is fragile or absent. A vial of factor VIII delivered to such a place may, by the time it reaches the patient, be biologically dead. The drug works in the laboratory and in the wealthy hospital. It frequently does not work at the point of use. In 2023, a research group at the University of Wyoming, led by the molecular biologist Thomas Boothby, published a paper in Scientific Reports describing what happens when factor VIII is dried in the presence of the tardigrade protein CAHSD. The CAHSD protein, the same intrinsically disordered protein that the lineage uses to vitrify its own cytoplasm during desiccation, was added to a solution of human factor VIII. The mixture was dehydrated. It was kept at room temperature. It was repeatedly rehydrated and re-dried. It was heated. It was stored dry for extended periods. In every condition the team tested, the factor VIII protected by CAHSD retained most of its clotting activity. The dried preparation, kept at temperatures up to 95 degrees Celsius, hotter than the inside of a closed car in summer, hotter than a large fraction of the conditions factor VIII is shipped through, remained functional. The cold chain in this preparation was no longer necessary. The implications, if the work scales out of laboratory conditions and into clinical and pharmaceutical use, are not subtle. A patient in a remote clinic with no reliable refrigeration could be given a stable dry powder, mixed with sterile water at the point of use, and treated. The same logic applies in principle to many other biological drugs, vaccines, antibodies, certain enzyme replacement therapies, cellular treatments. The cold chain that currently constrains where modern medicine can go is, in part, a consequence of the fragility of the molecules medicine depends on. The tardigrade has, for 500 million years, had a solution to that fragility. The solution is now beginning to be borrowed. Boothby's group did not stop with factor VIII. In 2024, the same lab, with the senior scientist Silvia Sanchez Martinez leading the work, published a study in Protein Science showing what tardigrade CAHS proteins do when they are introduced into ordinary human cells in culture. The result was unexpected even within the field. Under stress conditions, drought, certain chemical insults, the CAHS proteins inside the human cells did what they do in the tardigrade. They formed soft gels. The cell's metabolism slowed. The biochemistry that ordinarily proceeds at the rapid pace of a living mammalian cell decelerated, sometimes substantially, with the cells effectively entering a kind of induced biostasis. When the stress was removed, the gels dissolved and the cells returned to their previous metabolic rate without any obvious damage. This is, in conventional terms, a significant result. Mammalian cells are not, by ordinary biology, capable of entering controlled hibernation. The processes that allow ground squirrels to slow their metabolism in winter, or that allow bears to sleep through several months without eating, are complex multi-tissue affairs that are difficult to translate to single cells in a dish. The tardigrade's mechanism, by contrast, runs at the level of the individual cell. It does not require organ systems or hormonal cascades. It requires only the right disordered proteins and the right trigger. The implication is that with the right tools, human cells could be paused for medical preservation, for organ transport, conceivably for radiation protection during long-duration space travel, using a chemistry that the tardigrade has been quietly running in moss patches around the world since long before there were mammals. The radiation protection use case is not theoretical. In February of 2025, a team led by Giovanni Traverso at the Massachusetts Institute of Technology and James Byrne at the University of Iowa published a paper in Nature Biomedical Engineering describing what happens when Dsup, the original tardigrade DNA shielding protein, is delivered into mammalian tissue using lipid nanoparticles. The nanoparticles in question are the same general technology that delivers messenger RNA in modern vaccines, small fatty bubbles that ferry genetic instructions into cells. In this case, the instructions were the gene for Dsup. The cells receiving the instructions began to produce the tardigrade protein. The Dsup, made inside the recipient cells, found the chromatin in the recipient nuclei and bound to it. Then the team irradiated the tissue. In mice receiving the Dsup nanoparticles, healthy tissue around the irradiated zones, particularly the salivary glands and the lining of the gut, the tissues that suffer most during radiation therapy for head, neck, and gastrointestinal cancers, sustained measurably less damage than in control animals. The tumor cells, which had not received the protective treatment, took the full radiation dose. The healthy cells, with their tardigrade protein in place, retained much more of their function. In a clinical context, this is exactly the asymmetry that radiation oncology has been trying to engineer for decades. Roughly 95% of patients receiving radiation therapy for tumors in the gastrointestinal tract experience some level of treatment-induced damage to their healthy tissue. Reducing that fraction would change the lives of a great many cancer patients. The work, as of 2026, is in late preclinical stages. The first human trials are being prepared. Whether the approach will scale to clinical use, whether the protein will produce unforeseen side effects in human bodies over longer time periods than mouse studies can show, whether the manufacturing logistics can be solved, these are open questions, the way they always are at this stage of translational medicine. What is not in question is that a protein extracted from a small invertebrate that lives in damp moss is now sitting in regulatory pipelines as a potential cancer-supportive therapy. Dsup has also turned up in a different translational direction. In late 2025, a research group reported in Science Advances that expressing Dsup in the small nematode Caenorhabditis elegans, the laboratory worm that has been one of biology's most important model organisms for decades, extended the median lifespan of the worms. The mechanism appears to involve reduced oxidative damage to nuclear DNA over the course of the worm's life. The effect was modest, a percentage increase in median lifespan rather than a doubling, but the result is unusual. Genes from one phylum transferred into another and conferring not just stress resistance but extended longevity are not common in the literature. The work is preliminary. It will take other laboratories, other species, and several years of careful follow-up to know whether the effect generalizes. But the question is now on the table, in print, in a journal of reasonable repute. A tardigrade gene appears to extend the life of an unrelated animal. There is something worth noting in the accumulating shape of this work. The tardigrade was, until very recently, a curiosity, a small animal with strange physiology studied by a small community of specialists with no obvious relevance to human concerns beyond the general pleasure of knowing that something so small could survive so much. By the early 2000s, the curiosity had become an active research area. By 2016 and the publication of Dsup, it had become a source of mechanisms. By 2026, it has become, in a quiet way that has not yet fully registered in the broader culture, a source of medicine. The shift is not loud. There are no tardigrade-derived drugs in pharmacies as of this recording. The clinical trials are early. The cancer therapy is preclinical. The factor VIII stabilization is at the point where pharmaceutical companies are beginning to think about how to manufacture it at scale. The biostasis work is at the cell culture stage, with whole organism applications still a decade or more away. None of this is today sitting in a typical doctor's office or pharmacy, but all of it is in motion. The pipeline that runs from a moss sample to a research bench to a clinical trial to a treatment is, for several pieces of the tardigrade's molecular toolkit, already a pipeline with motion in it. The animals that produce these proteins do not know, of course. The Hypsibius exemplaris in a culture dish in Wyoming is being maintained by graduate students who feed it algae and check its hydration. It is laying eggs, eating, occasionally entering a ton when conditions in the dish go sideways. The Rhamazottius varionatus in Tokyo continues its similar small life. Neither animal has any awareness that, in research buildings on 3 continents, copies of their proteins are being produced, purified, and tested in the cells of a different vertebrate that towers somewhere around 3 orders of magnitude larger than they are. The asymmetry is total. We are paying very close attention. They are not. There is in this a particular kind of relationship that biology has not seen before. Domestication, the long process by which humans have shaped wolves into dogs and aurochs into cattle and grasses into wheat, is one kind of relationship, slow, mutual, involving changes in both species over generations. The tardigrade situation is not domestication. The animal is not being shaped. Its biology is being read, copied, and used. The proteins extracted from its cells are being put to human purposes that have nothing to do with the animal's life or evolution. The animal continues, unmodified, in its moss. The relationship runs entirely in one direction. The tardigrade gives, we take. The tardigrade does not notice that it has given. Whether this counts as a relationship at all in any moral sense is a question moral philosophy has not particularly addressed. Most of the frameworks that consider obligations to animals were built around creatures that can suffer in recognizable ways. Vertebrates with nervous systems substantial enough to support something like pain, animals whose welfare biology has worked out in some detail. The tardigrade does not fit those frameworks well. It has 200 neurons. Whether it can suffer, in any sense the word usually means, is unknown and probably unanswerable. The moral status of taking a protein from such an animal and putting it in a cancer patient is, at present, a question without much philosophical work behind it. The work, perhaps, will come. In the meantime, the animal walks in moss. The protein, copied from its body, sits in a vial in a lab freezer. The haemophilia patient, who will eventually be treated with a stable dry powder, has not yet been told that part of the formulation came from a creature 0.5 millimetre long that was, several 100 million years before any of this, learning how to dry up and come back from the dry. The patient will, in all likelihood, never need to know. The story, on this end of it, has the quiet, slightly improbable quality that accumulates around situations where something very small turns out to matter very much. The moss is still there. It has not, of course, gone anywhere. The patch on the wall outside the house, the cracks between the paving stones, the shaded hollow under the tree where the ground is always slightly damp, these were there before this recording began, and they will be there when it ends. The animals in them have not, during the time it has taken to describe them, stopped doing what they do. The stylets have continued to puncture cells. The bodies have continued to fold and unfold according to the moisture conditions of small spaces. Several thousand individual tardigrades have, somewhere within a few kilometers of where this is being recorded, completed a full cycle of drying and rehydrating without anyone noticing. The world above them has remained almost completely unaware of any of this, and that is, in the most exact sense, the way it has always been. Most documentaries about animals end with conservation. They arrive in their final minutes at the question of whether the species in front of the listener is going to survive the century, what threats it faces, what humans might or might not do to help. For most species, this is a reasonable framing. For the tardigrade, it is not. There are roughly 1,500 described species of water bear, and many more that have not yet been formally named. As a phylum, the lineage is in no danger from anything humans are currently doing to the planet. They live too densely, in too many places, on too many continents, in too many habitat types. They are too small for most of the things our species has done at our scale to register at theirs. Climate change will reshape some local communities, particularly cold-adapted endemics on the Antarctic Peninsula, on alpine moss meadows, in high Arctic soils, and some of those species may decline or disappear. The lineage as a whole will not. The water bear has, by now, lived through 5 mass extinctions. It is not, in any meaningful sense, at the mercy of a 6th. What this means for the conservation conversation is that the framing inverts. The question is not whether our species is going to allow the tardigrade to persist. The question is, much more interestingly, what the tardigrade can offer ours during the period that ours turns out to need offering. The relationship runs in one direction. The lineage that has spent 500 million years walking through damp moss is now in laboratories on 3 continents beginning to be borrowed from. None of the borrowing harms the lineage. The lineage continues to exist in moss undisturbed, while a small fraction of its molecular toolkit is copied into the medical pipelines of a species it has no awareness of and no relationship with. A great deal about that toolkit is still not understood. The molecular biology of cryptobiosis in 2026 is only partially worked out. Whether the dehydrated tardigrade is, at the molecular level, in a true vitrified glassy state or in some other condition that resembles vitrification, only superficially is a question still being argued in the literature. The exact mechanism by which Dsup protects DNA in some cell types while harming it in others has not been settled. The full catalogue of bacterial and fungal genes that have been transferred into the tardigrade genome over the lineage's long history is still being compiled, and each of them, like the dopa dioxygenase from the small population in Hainan, may turn out to encode a chemistry the lineage has put to unexpected use. There are tardigrade species that have barely been studied at all, the marine ones especially, mostly known only from their morphology, with their molecular biology still unexplored. The lineage is large, the work is slow. The funding for studying small invertebrates is, in the modern research economy, modest. The work continues anyway, in places that include both the obvious and the surprising. The Boothby lab in Wyoming continues with the CAHS proteins. The Concordate group in Paris continues with TDR1 and its cousins. The Lee group in Qingdao continues mining the genome of Hypsibius henanensis. And in May of 2025, on the Axiom 4 mission to the International Space Station, the Indian Institute of Science in Bangalore sent up a small population of paramacrobiotus tardigrades collected from a moss-covered concrete wall on its own campus. The astronaut Shubhanshu Shukla, on India's second human spaceflight, carried them aboard. The animals were exposed to several weeks of microgravity and orbital radiation, and the question being asked is the one Boothby's group asked years earlier and the one the TARDIS mission asked before that. Which genes turn on, and what do those genes do? And what does this animal know how to do that the rest of us do not? The data is still being analysed. There is also, gathering quietly behind the laboratory work, an astrobiological angle. The tardigrade has become, in the planning documents of various space agencies and academic programs, the standard reference for what kinds of biology might be possible elsewhere in the solar system. Proposed missions to Europa, the icy moon of Jupiter, whose subsurface ocean is one of the most intriguing astrobiological targets we know of, often cite tardigrade physiology when describing the kinds of organisms that might, if life is there at all, be detectable. The same is true for missions to Enceladus, the ice-shelled moon of Saturn that vents geysers of liquid water into space. The same is true for various conceptual studies of subsurface life on Mars. The water bear has, almost by accident, become a benchmark. Not because anyone expects to find tardigrades on other worlds, but because the lineage represents a working answer to the question of whether life can persist in conditions that include long dry periods, freezing temperatures, vacuum exposure, and high radiation. The answer on Earth is yes. The answer elsewhere in the solar system may also be yes. We have at least one confirmed example of a planet where this has happened, and the example is walking in the moss outside. What the tardigrade teaches, beyond the specific mechanisms it has worked out, is something about what the boundary of life can look like. For most of the history of biology, certainly for most of the history of the way ordinary people have thought about biology, being alive has been understood as a continuous condition. An animal is alive or it is dead, and the line between those two states is one of the most consequential lines in nature. Most life forms cooperate with this framing. They are alive while they are alive, and once they cease being alive, they do not come back. The tardigrade does not cooperate. The tun state is, in any biology textbook framework, neither alive nor dead. It is something the framework was not designed to accommodate. A frozen tardigrade in moss in Antarctica is not metabolizing, not aging, not interacting with its environment in any active sense, and not also decomposing. The chemistry that ordinarily defines the difference between organic life and the inert world has been, by the lineage's molecular machinery, quietly suspended. The animal can be held in this state for years, decades, possibly centuries, and then, given water, can rejoin the world of the living without any obvious gap in continuity. The category of being alive turns out to be more porous than the framework suggested. It is the kind of fact that, if it had been discovered in some exotic deep-sea organism almost no one had ever heard of, would be classed among the strangest things in biology. It has been discovered instead in the most common animal in any patch of moss in the temperate world. The strangeness is widely distributed. It lives in any garden. There is a way of looking at this animal that, while not strictly scientific, is also not in the end untrue. The tardigrade is not, in any meaningful sense, a witness to the deep history of life on Earth. It does not perceive what is going on around it at the scale on which deep history operates. It cannot remember the Cambrian seas. It cannot remember the asteroid that ended the dinosaurs. It cannot remember the long, slow rise of the mammals or the brief late arrival of human beings. The lineage has lived through all of these, but no individual water bear has experienced any of them, and the lineage itself does not have anything we would recognize as memory. And yet, the body the animal in the moss this morning is using, the 8 legs, the stylets, the disordered proteins, the borrowed bacterial genes, the DNA shields, the cryptobiotic switch, is the same body, in essentially the same form, that walked on damp ancient surfaces before there were forests, before there were vertebrates, before there were almost any of the species the listener of this documentary could name. The lineage is older than memory, older than history, older than almost everything the human imagination ordinarily reaches for when it wants to feel the depth of time. The pyramids are about 4,500 years old. The first cities are around 10,000. The first agriculture is around 12. The deepest cave paintings are 40. The deepest known human bones are a few hundred. The tardigrade is 500 million. Not 1,000 times older than the human species, 1,000,000 times older than the oldest stories the human species has ever told. The body in the moss this morning is, in the most literal sense the language permits, ancient. It is also, in the same body, walking. It is doing both things at once. The continuity of the lineage is, in the end, the most remarkable property the animal possesses, and it is the property the animal is least equipped to know it possesses. The end of a documentary about an animal usually wants at this point to do something, to gesture toward the future, or to summarize what has been learned, or to draw a moral. There is no moral here. The animal has no moral. The animal is small and slow and transparent, 0.5 millimetre long, walking across a wet leaf. None of what has been said about it changes what it is. None of what has been discovered about it has, in any sense the animal can register, reached it. It is morning somewhere. The moss has been wet for about an hour. The patch on the north side of the wall, the patch in the courtyard, the patch on the gravestone, the patch in the high country 100 meters above any nearby town. All of them, in their slightly different conditions, contain animals that are unfolding now, beginning to walk, finding the first soft cell of the day to drink from. Inside laboratories on 3 continents, copies of these animals are being studied. In a culture dish in Tokyo, the descendants of SB1 are continuing their small lineage, generations now removed from the moss in Yukidori in 1983, none of them aware that one of their ancestors was famous. In a freezer in Wyoming, a vial of CAHSD protein, extracted from cultured tardigrades, sits among other vials of biological reagents, waiting for the next experiment. In a regulatory pipeline somewhere between Boston and Iowa, the Dsup mRNA preparations are moving slowly through their preclinical paperwork. In a crater on Mare Serenitatis, the bodies of several thousand tardigrades, neither alive nor in the cryptobiotic state that allows revival, sit in slow lunar weathering, and the animal in the moss continues. The morning's drinking proceeds. The legs move slowly. The body is 0.5 millimetre long. It does not know about Tokyo. It does not know about Wyoming. It does not know about Boston. It does not know about the moon. It does not know about you sitting wherever you are sitting, hearing this. You, by now, know about it. Somewhere in a garden within walking distance, in moss you have probably passed without noticing more times than you could count. Several thousand of these animals are doing exactly what they have been doing for 500 million years. They are folding when the moss dries. They are unfolding when the rain comes. They are moving forward slowly on their 8 legs, with their stylets ready and their proteins held in place into the next small interval of damp survival. When you walk past the wall tomorrow morning, the moss will be there, the animals will be there, the lineage will be there. None of them will look at you. You can, if you feel like it, look at them. There is no obligation. The animal has been doing its work without anyone watching for 500 million years. It does not need anyone to watch now. Still, tomorrow morning, on the way out of the house, on the corner where the wall meets the path, on the cracked stone where the moss has been growing for some long time you have not measured, there is a creature there, or 2, or 40. You will not see them. They will not see you. Their eyes are not built for that. But the lineage they belong to has been on this planet for longer than yours has been on it, by a very large factor. They were here first. They will be here last. The morning, for whatever it is worth, will belong to all of them at once. They will not notice. You now know. Good night.